All right, this is Spencer and Brady doing lesson 13 problems. I'm going to go ahead and get 1 and 5, and Brady's going to get in the middle. He'll get 2, 3, and 4. So let me just go ahead and get this screen share set up. All right, so number 1 is asking, you wish to use DNA sequencing to determine the nucleotide sequence of the DNA molecule shown below. And it gives us our strand right here, 5 to 3 prime. And then it says the primer annealing site indicates the region of the template DNA strand where the primer will anneal during the sequencing reaction in order to generate the newly synthesized strand. Note that the newly synthesized strand will be complementary to the sequence above. After DNA sequencing using the Sanger method described in class, the DNA fragments were separated by electrophoresis on a gel. So A is asking what would the banding pattern look like, and it tells you how to set it up using the different DT and TPs. And so basically, all you have to do is we have our strand given to us 5' to 3', and negative is going to be up top, positive is going to be below, so 3' and 5' like I have marked here. And you can start really at either way and fill out your table that way. And so if I started at 3', we do G, and complementary of G would be C, so I mark that down. And then complementary of A is T, so you just keep moving up, going 3' to 5' or 5 to 3. I mean, it really doesn't matter how you write it down. And that's all you have to do for that. You just use the strand that they gave you. And then it says, for parts B through D of this problem, suppose I try to determine the nucleotide sequences of 3 other template DNA molecules. However, I make a mistake during each of the 3 reactions. Please describe what will happen in the reaction with each mistake. And so for B, suppose I use E. coli DNA polymerase 1 during the sequencing reaction instead of the Taq polymerase or TAQ. And this wouldn't synthesize because it would denature, it's too hot. Whereas TAQ polymerase, it's able to synthesize under the high heat, which we're given in the notes is 72 degrees Celsius. C, suppose I accidentally add E. coli DNA helicase to the reaction in addition to the normal DNA sequencing reaction components. The reaction should still work. Nothing's going to stop it. Helicase ain't going to impede anything. D, suppose I add DT, ATP to one of the 4 tubes. However, I forget to add the other 3, GTP, TTP, and CTP to the other tubes. Assume that the other components used for sequencing are normal. So basically, complementary to ATP would be TTP. And so we know that there would be a premature termination at the first T nucleotide. All right, go ahead, Brady.
Oh, sorry, I fell asleep. Just kidding.
So funny.
All right, number 2, filling in the blanks. A, during a typical PCR cycle, the DNA synthesis step is done at 72 degrees Celsius, while the primary annealing step is done at 55 degrees Celsius. You know,
hey, let's get that screen share going.
Yeah, I'm not screen sharing, so let me hurry and screen share. Sorry, y'all, you got to see my ugly mug. Okay, is it working now? Yep. Yeah. Okay. Now B is AMPR is a common selectable marker that is found in many plasmid vectors, while restriction endonuclease recognizes and cleaves DNA molecules at palindromic sites. It's good enough for me. C is during agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates toward the positive electrode during the electrophoresis. It goes away from the negative. D, DDATP lacks the 3-OH group and the 2-OH group. E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. And F, during blue-white screening, IPTG is commonly used to introduce the expression of the lac operon, while XGAL is cleaved by that giant word that I'm not even going to attempt to say, beta-galactidase, to turn E. coli cells blue. So that's number 2. Now number 3 says you're interested in using PCR to amplify the region of a double-stranded DNA fragment from E. coli. The sequence to be copied is located between the sequences in the bold below. So right there, the bold kind of gives it the first one away. It says, write the single-stranded DNA sequences that you would use for the 2 primers used in the PCR experiment. Know that each primer should anneal to one of the sequences in bold. So primer number 1, I copied and pasted from the 5 prime. It looks like I actually turned that one into primer number 2, and this one's primer number 1. But they're both at the 5 prime end on each one of those. So there they are. The mathematical equation that's used to determine annealing temperature is right here. For one primer, we get the number 56 to plug in. For another primer, we get 50 to plug in. And those are counting the GCs. And when you do that, you're going to end up with 62.3 degrees Celsius and 59.9 degrees Celsius. Or 8. I think it's supposed to be 8. I might have put 9. Fix that. Okay, now C, each PCR cycle contains a DNA synthesis step. What are the sequences of the 2 newly synthesized strands of DDC-TP is accidentally included in the PCR experiment? Assume that ATP, TTP, and GTP are all other appropriate PCR components are found in the reaction mixture. And so this is what you'll come up with. You'll end up with primer 1, 5 prime to 3 prime, GGGA, TTA, GC, and I won't repeat the second one because that's painful for your ears. Then D says, when we learned about DNA replication in E. coli, we learned about the DNA, and damn, why are these 2 proteins not included in the PCR experiment? What I decided to put was that DNA A catalyzes, and the DAM controls when replication is occurring. Forgot to put another A in there too. Okay, so hopefully that satisfies that one. And then 4, if I can find it. I don't know where it is. This is probably it. It's just asking to map all these things out, this EcoR and the Hindu 3 and stuff like that, Hin3. And so for this, I drew a picture. Ignore that, that's number 1. So this is 4A. I just made a chart starting at 100, going to 1,000. I should have made it one bigger, so my 900 was in there, but that's all right. We didn't have a 900. And so you just follow your numbers down, put the X's in the box. I mean, these aren't perfect because my drawing skills are not that great, but they're very close, I guess. B, then you're just going to do the addition thing. Sister Mayo made a video that explains this stuff much better than I'm going to explain it, but as long as your numbers add up correctly, then you know that you have your right, what am I going to call these things? Spencer, chime in. What are they called? Reaction enzymes?
Yeah.
Then you know if you have the right reaction enzymes in their correct spots, as long as they add up. So, and then C just goes from least to greatest, starting with 50 and goes to 500. So it'd be 50, 100, 150, and 500 base pairs. And Spencer, do number 5.
All right. So basically, we got number 5 straight from the notes. Let me get to it real quick. Okay, so number 5, you're interested in studying a gene called your favorite gene that you have identified in the yeast Saccharomyces. Detail the steps that you would use to generate the cDNA. Saccharomyces mature mRNA, clone a cDNA version of YFG into a plasmid vector and use blue-white screening to identify plasmids that contain the YFG cDNA. So from the notes we were given these steps. So for producing and cloning, we're going to start with purifying the mature messenger RNA from a sample of cells. You mix it with a primer, and then you add reverse transcriptase and DNTPs to synthesize the strand of DNA. The new strand of DNA is complementary to the original messenger RNA that we used. And then we add RNase H to digest the majority of the messenger RNA. And then that's just like a side note from it is one consequence of the RNase H treatment is that the messenger RNA is degraded to produce the primers for DNA synthesis. And then after that, we just have to add a DNA polymerase 1 to synthesize the second strand. So that's how you do that part. And then for the blue-white screening, you just use the beta galactosidase. And if the colony is blue, like it says right there, it converts the X-gal into a blue product. If the colony is white, that's because the galactosidase cannot convert X-gal into a blue product. So we pulled those from the notes, and that's how we did number 5. Now, well, that was the last video, so that's pretty awesome. Checking out for the semester. See ya.